Hello and welcome back to Survey of Biology as we begin Unit 9 on cell division. So here we'll be looking at the cell cycle, the steps that a cell goes through during its life leading up to division, mitosis and meiosis. So you can see from the pictures here, the upper left is showing a quick kind of color-enhanced steps of mitosis, where we go from having one mother cell to two identical daughter cells. The other two diagrams are showing the last stages, cytokinesis, when these two daughter cells are actually splitting apart to become two brand new cells. So the biggest question we need to look at when we talk about cell division is, why do cells need to divide in the first place? And the first is reproduction. We need more cells. And we're going to look at something called binary fission, which is another way of talking about mitosis. And what this does is we start with one cell right here, and we end up with two daughter cells that are genetically identical to the original mother cell. And that's a big thing to remember, is that we start with one mother cell, we end up with two genetically identical daughter cells. The second reason to divide is growth. We need to increase the cell number. So we all start as very small babies, and we're much larger now. We have more cells in our body, so we need to increase the number of cells. The third reason is tissue renewal. So here's replace and repair damaged tissue. So if you get a cut, you may need to grow new skin cells, new muscle cells, whatever it might be. And the last one is to increase something called the surface area to volume ratio. Every cell has to take in nutrients through its cell membrane and get rid of waste. If the cell gets too big, the surface area to volume ratio becomes too small, so the volume of the cell is much too large to take in what the cell needs and get rid of the waste. So the cell will divide to become more efficient. So before we get into the actual division in the cell cycle, let's take a quick look at DNA. So in each of our cells, every single body cell has a complete set of what's called the genome. That's the total DNA of a cell. Everything about us is in every single cell. And for a human, that genome is 46 chromosomes. Two sets of 23. One is maternal from mother, one is paternal from the father. And so in each of our body cells, so skin cells, muscle cells, nerve cells, those are called somatic cells. We have a complete set of 46. And that's what's called diploid. Sometimes it's referred to as 2N because it's got two sets of the chromosome. We have the two sets of 23. And the vast majority of our cells have that diploid number 46. Now in humans, the gametes, the sperm and the egg, the sex cells, are what's called haploid. They only have one set, so they have 23 chromosomes, and we'll get to that when we talk about meiosis and how 23 can get back to 46 through the process. So we just need to do a little bit about DNA. And so if we have a picture of our DNA, and we stain it, and we get what's called the karyotype. And what we're looking at here is a karyotype of an individual, where we can see that there are 23 pairs. So you can go right through, 1, 2, 3, all the way up to 22. Now the 23rd pair are the sex chromosomes. And in this case here, we've got one X and one Y. So this individual here, because of that, would be a male. If it was XX, it would be a female. But the karyotype gives us a picture of a human's genome, what their DNA actually looks like. Now, most of the time, that DNA is in the form of what we call chromatin. And the way I remember chromatin is long and thin. So this is uncondensed DNA. So if you were to pluck off a skin cell, chances are the DNA in that nucleus would be invisible beneath a microscope. It hasn't condensed yet. And we'll need something to happen before we can actually get a look at that. And that's what happens when chromatin turns into chromosomes. So chromosomes is DNA, and it's complex with proteins to help maintain the structure. It helps hold it together and make it stable. And when the cell starts to replicate, it condenses to form something called sister chromatids. So if you look down at the picture on the lower right-hand corner, down here, what this is showing here is a chromosome that has been replicated. So it's a copy of itself. And they are attached at this region called the centromere. So this is going to be a very important structure when we talk about mitosis and meiosis, is that when the DNA replicates, it forms these sister chromatids that are attached at the centromere region. So a quick look of how we get from the chromatin to the sister chromatids. So over here on the right, here are sister chromatids. It looks like an X. Okay. So we start off over on the left with a chromatin that's long and thin, and what happens is that DNA gets wrapped around these proteins called histones. And all that wrapping and looping causes the DNA to condense down, and that's the structures we get that we call sister chromatids that, again, that are kind of attached to each other at the centromere region. So here's what it would actually look like in a cell. So the cell on the left is in what we call interphase. It's not getting ready to divide yet. So it's kind of undifferentiated. We can't really make out DNA in there. But if you look at that cell right in the middle, it's slightly larger, and there's these snake-like structures. Those are the chromosomes that have condensed down, and the cell is getting ready to divide. So here's a quick chart that kind of outlines the differences between the two. So over on the right, the chromatin is unwound. It's found throughout interphase, and it's DNA that's being used for macromolecule synthesis. So what does that mean? Is that DNA is providing the blueprint to make molecules like proteins that can serve as enzymes and help with metabolism. Chromosomes, on the other hand, over on the left, are tightly packaged DNA. They're found only during cell division, and we're going to look at it right away as mitosis. And then the DNA is not being used for macromolecule synthesis. We're not copying it. We're just getting ready to kind of divvy it up between different cells. So this diagram here, so if you start in the upper left, so there's a chromosome that's been condensed. Right here, it's not a sister chromatid yet. During what we're going to call the S phase synthesis of mitosis, we make a copy of it. That's where we get the sister chromatids. So they're held together at this centromere region, and eventually the sister chromatids are what's going to be divided, pulled apart to give the DNA to the new daughter cells. And here's the actual cell cycle that we get to. So the cell cycle is a series of events that takes place in a cell that leads to its duplication and division. And again, as I said before, it's taking one mother cell and giving us two identical daughter cells. And if we look at it, we're going to talk about mitosis eventually, but mitosis is a relatively short part of the cell cycle. The cell doesn't spend a lot of time in it. If we want to look at the longest phase or the longest stage of the cell cycle, it's this phase called interphase. So most of what a cell does happens during interphase. So if we start right at the beginning of interphase, what we'll see is this phase called G0. Okay? And G0 is where the cellular contents, excluding the chromosomes, are duplicated. So G1 actually stands for gap 1, but I like to talk about it in terms of growth. So it grows, it gets nutrients to grow, and it's just kind of carrying out its life cycle. Now, as it gets ready to divide, a decision has to be made. And sometimes that cell will decide not to divide. And if it does, it enters this phase called G0. And here's where we kind of have mature cells. Here's cells that are carrying out their function they're supposed to. So a skin cell, a nerve cell, a muscle cell. But if the cell gets a signal that it needs to divide, it will go through G1 and into S, which stands for synthesis. So here's where the DNA, the chromosomes, are duplicated. So we've got our 46 chromosomes. Each one of those is going to be copied so that we've got twice as much DNA. and you can see the little sister chromatids in the picture right there. After the S phase, we go into what's called the G2 phase, and here the cell double checks for duplicated chromosomes, for any errors, makes needed repairs, and it just kind of grows and gets ready, and that's when it will actually finally enter mitosis and split into two. So as you can see, most of the cell cycle actually takes place in interphase and is growth, preparation, and synthesis of the DNA. So the last thing we'll talk about real briefly is in the cell cycle, as we go through interphase, there's these places called checkpoints. And these checkpoints are places where if they don't make it past, the cell cycle can stop and the cell may eventually die. And you can see on the diagram here, there's 1, 2, 3, 4, 5, 6 of them. And so there's a lot of different places where the cell kind of checks. Does it have enough DNA? Does it have enough organelles? Are the spindle fibers ready to go? And goes through and can stop the cell cycle if things aren't perfect. And that's one of the reasons why we don't see as many mutations as we might normally, because cells that have problems are prevented from actually dividing before they get there. And so I just want to talk about a couple major checkpoints. The big one being this one down here called the G1 checkpoint, and it's also known as the restriction point. Okay? When it passes this, if it doesn't pass, it goes to G0 and it's a mature functioning cell. But if it passes G1, the cell is committed, let's see if I can fit it in there, committed to divide. And it's going to go through, and as long as it makes it past the other checkpoints, is going to get there and divide for that. So all you have to remember about a checkpoint is they are places where the materials of the cell are checked to make sure everything is necessary to go on to the next step. And so now, I think what we're going to do is take a quick look at an animation about how the cell cycle works.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into 3 discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell.
And I think we'll pause it right there because we're getting a little ahead of ourselves into our next section, which will be mitosis. So that wraps up the cell cycle. Thanks for your time, and we will talk about mitosis in our next one.